Michael Levin's Xenobots are synthetic life forms created from the cells of the African clawed frog, Xenopus laevis. They are not traditionally engineered robots but rather biological entities designed by assembling living cells into specific configurations using computational models and bioengineering techniques. Michael Levin and his team developed these Xenobots to explore principles of self-organization, collective behavior, and the potential for novel biological systems. Key points about Xenobots. Composition. Xenobots are made from frog skin and heart cells. The skin cells provide structural support, while the heart cells' contractions enable movement. Design. Their shapes and behaviors are designed using evolutionary algorithms, which simulate natural selection to optimize for specific tasks, such as locomotion or object manipulation. Self-organization. Xenobots demonstrate emergent behaviors, such as collective movement and problem-solving, despite being simple in design. Reprogramming. Levin's work emphasizes the plasticity of cellular systems, showing that cells can be reprogrammed to adopt new functions and behaviors outside their original context. Why Xenobots do not confirm the roles of genes and evolution from the standard perspective. Top-down design. Xenobots are not products of natural evolution but are designed by humans using computational tools. Their creation relies on external intelligence, scientists and algorithms, rather than the blind, undirected processes of mutation and natural selection. Predefined goals. The design of Xenobots is goal-oriented, aiming to achieve specific functions. In contrast, evolution has no foresight or goals. It operates through random mutations and environmental pressures. Gene expression versus novelty. While Xenobots rely on the genetic machinery of frog cells, their behaviors and structures are not directly encoded in the genome. Instead, they emerge from the interaction of cells in novel configurations, highlighting the importance of cellular plasticity and environmental cues rather than strict genetic determinism. Evolutionary theory. The creation of Xenobots does not challenge the core principles of evolution, e.g., genetic variation, selection, and adaptation. Instead, it demonstrates how cells can be repurposed in ways that evolution might not explore naturally, given the constraints of historical pathways and ecological niches. Broader implications. Levin's work with Xenobots highlights the importance of understanding biological systems beyond the gene-centric view. It emphasizes the role of cellular communication, how cells interact and coordinate to achieve complex behaviors. Environmental cues, how physical and chemical environments shape development and function. Plasticity, the ability of cells and tissues to adapt to new contexts and roles. In summary, while Xenobots are a groundbreaking achievement in synthetic biology, their creation does not confirm or refute the roles of genes and evolution. Instead, they expand our understanding of how life can be manipulated and repurposed, offering insights into the flexibility and potential of living systems.